Go. So, as Sarah Jane said, I'll be sharing with you on our insights into the role of the immune system in neurodevelopment. And as she mentioned, I'm a paediatric neurologist here at Starship Children's Hospital, and I've been very privileged to receive the Neurological Foundation Senior Clinical Research Fellow Award. So I'm going to start by way of a background and introduction, then talk about some of the clinical experience that I've had, and then what to next with the project grant, again generously sponsored by the Neurological Foundation. And so when we look at neurodevelopmental disorders, we're looking at a wide range of disorders that are generally overall quite common and affect 1 in 6 children. Perhaps most well known to many of you will be the autism spectrum disorder, in which case there is social communication difficulties and problems with restrictive and repetitive behaviours. And autism spectrum disorder affects 1 to 2% of the general population. Attention deficit hyperactivity disorder affects up to 5% of the population and is associated with inattention, hyperactivity, and impulsivity. Tic disorders and Tourette syndrome are movement disorders involving the sensory motor circuits, and these affect up to, transient tics affect up to 20% of children, but Tourette syndrome would affect 0.5 to 1% of the population. And childhood obsessive-compulsive disorder, which is seen in about 30% of children with Tourette syndrome and can also overlap with autism spectrum disorder, is associated with intrusive obsessional thoughts that then require compulsive action to alleviate the distress. And here's a video of a boy that we looked after in Sydney with Tourette syndrome, shared generously with permission. And you can hear these repetitive vocal tics and motor tics as well, which, as you can see in a severe form, are very disabling. And so the prevalence of neurodevelopmental disorders overall is on the rise. And this is a graph taken from the National Health Survey in the United States, which shows an increase over the past 20 years. Just of note, this graph does include intellectual disability and cerebral palsy and hearing loss, so other conditions as well, which I won't be talking about today. But if you looked at a graph of autism spectrum disorder or ADHD, you'd see a similar rise in prevalence over the past few years. Now, this clearly could be related to a change in diagnostic practice, and it does to an extent. The question, for example, used in the survey for the data here changed in 2014 from autism to autism spectrum disorder. So there are those factors at play. But I don't think that, and you could debate this, but I don't think that it fully explains the rise in prevalence over the last few decades. Obviously, such a quick rise as well, could it be explained by genetics? And this raises the question, are there environmental factors involved in the rise in prevalence of neurodevelopmental disorders over the past few decades? And so what about, could one of those environmental factors be, in fact, be inflammation? And the interest in, specifically, inflammation in pregnancy. And the interest in inflammation in pregnancy dates back to the middle of the last century, when there was a significant rise in autism and schizophrenia associated with the 1964 rubella pandemic. Now, some of you may be aware that rubella crosses the blood-brain barrier and actually can infect the brains of children. So you could say, well, perhaps this was a subclinical infection in the offspring, in the babies in utero. But maternal influenza has been shown to have the same association with neurodevelopmental and neuropsychiatric disorders in offspring. And the second data point here refers to a nationwide study looking at the serology of mothers who had influenza in pregnancy, and there was a 7 times the risk of schizophrenia in their children. Thirdly, maternal infection generally has been associated with a rise in autism spectrum disorder as well. And the fact that there are many different infections that have been associated with a rise in neurodevelopmental and neuropsychiatric problems raises the question, well, actually, is it the inflammatory response? And so then there has been studies looking into inflammatory conditions in pregnancy. And perhaps one of the conditions which is the easiest to study in some ways is maternal autoimmune disease, because these are chronic pro-inflammatory conditions where the immune system is dysregulated and affects self. And so maternal autoimmune disease has been associated with a 29% increased incidence rate of Tourette syndrome in male offspring, 30% increased odds of autism spectrum disorder, and 30% increased rate of ADHD as well. And so one model that could explain this, and this is a model that has been thoroughly studied in animals but still lacks a lot of data in humans, is that perhaps immunological activation in pregnancy, whether it be infection or provoked by infection or autoimmune disease, has an effect on the fetus, whose then response depends on their immune system and their genetic vulnerabilities, which may prime them to be at increased risk of other second hits throughout development, such as, for example, in adolescence, stress and drug abuse, so that they have a higher risk of psychiatric and neurological disorders. And so one way to study this is to look at animal models. And so how this has been studied is that there are bacterial mimetics like lipopolysaccharide and viral mimetics like poly-IC that stimulate an immune response in mice or rodents, or there have been studies in monkeys as well. And then looking at the neurodevelopment of the offspring. And the effects of inflammation in pregnancy are quite diverse actually, and they've looked at it on many different levels. For example, if there's been mothers had a viral infection or immune activation in pregnancy, then the offspring mice have a change in the volume of their white matter and their grey matter. Another way that things are changed is that looking at the neuronal circuitry that can be altered as well in neurochemistry. And also the immune activation in utero affects the microglia as well, which we're becoming increasingly aware of has an important role in neurodevelopment too, which I'll go into detail shortly. And then these changes that have been seen in the mice offspring are linked with changes in mouse behavior as well, such as abnormal vocalization, so a lack of intonation or change in the quality or the quantity of the vocalizations, repetitive behavior, such as they studied this by looking at how mice bury marbles repetitively, and impaired sociability, how interested are the mice in novel objects. And so this is one example of one of those mouse tests called the open field exploration test. So they exposed one of the children who was one of the baby mice, that were exposed to inflammation in pregnancy into an open area that is unfamiliar to them. And the mouse born to the sham infected mother, so this mother had the was anesthetized as if they were being infected, explores the whole area. And as you can see, the whole grid and the little circles are where the mouse goes up on its hind paws. A mouse born to a virus-infected mother would only roam around a very small proportion. It would move just as often, but it explored to a much lesser an extent. And these same findings have been found with not virus-infected mothers, but also bacterial and using those bacterial and viral mimetics that I mentioned earlier. And so what about that microglial mechanism? How does inflammation in pregnancy appear to affect microglia from looking at these animal models? And so just to go back, microglia are the white blood cells of the brain, and they comprise 20% of brain cells. And they enter into the central nervous system or the brain, forming brain quite early on in the first 8 to 9 weeks of gestation. And they're important in a number of different areas. They are involved in apoptosis or programmed cell death of neurons. They produce factors which promote neuronal growth and also the growth of other glial cells or structural cells of the brain. They're involved in neuronal maturation and differentiation as part of development, and also the maturation as well of the structural brains of the cell or the oligodendrocytes, which produce myelin too. And so this graph just shows you an overview really of the neurodevelopment and immune system development side by side. So you can see here neurogenesis, inflammation of new neurons and glial or structural brain cells occurs quite early on. And there's the neuromigration. Apoptosis is programmed cell death as the through the neurodevelopmental process, and then synaptogenesis, is the formation of synapses between the neurons. As mentioned, the glia enter the CNS quite early on, or the central nervous system quite early on, around 8 to 12 weeks, and then there's the maturation of the innate immune system, or that early immune response too, after birth. And so these graphs here are just to show how closely associated the microglia are in synaptic functioning. So these are glutamatergic nerve synapses. So this is the presynaptic neuron sending the message to the postsynaptic neuron. This here is an astrocyte, which is one of the structural cells of the brain, and this green cell here is the microglial cell. So just to show that the microglia are involved in strengthening synapses and also in pruning synapses as well. And this graph or figure on the right shows that should be MHC1 actually, which is an immune molecule which is expressed in synapses as is also important in synaptic pruning. So just showing how the immune system overlaps with neurodevelopmental processes. This is another model mostly shown through animal models as well, and actually initially adopted from adult disease research, which shows that a naive microglial cell, when exposed to inflammation, should produce a normal inflammatory response. One hypothesis of how systemic inflammation in pregnancy might affect neurodevelopment and later on is that when the microglia are primed by being exposed to an abnormal environment in utero, then with a second hit or coming across systemic inflammation again, they produce an exaggerated inflammatory response. And so this has been shown, but not necessarily across the course of neurodevelopment. So what can we see about how important the microglia might be in neurodevelopment in humans? And so this is a study that looked at the brains of 9 people who had Tourette's syndrome compared with 9 healthy brains, and the relevant part of the brain, the striatum, to Tourette's syndrome. And what they looked at was how active the genes were in that part of the brain in the people with Tourette's syndrome, which genes were most active, in the people with Tourette's syndrome compared to neurotypical individuals. And the genes that were less active in the Tourette brain were the neuronal genes, which is what you would expect, because these are the genes that, if you look at big genetic studies, appear to be affected in Tourette syndrome. The genes which were more active were the immune genes and the microglial genes, showing that there was a degree of microglial activation. What we don't know is, because these were adult brains, was the microglial activation involved in the process of the Tourette syndrome, or perhaps is this a secondary response to what's been going on in the neurons? And that's something we don't know yet, but clearly there is an immune response there. Similar pattern is seen in the brains of children with autism, or not children, but adults with autism. So, or autism brains. So here, the less active genes are shown in green, and the compared with the controls, and the more active genes in the autism brain are red compared with controls. So the less active genes are the genes which are involved in synaptic processes, the neuronal functioning, neuronal projection and synaptic vesicles, so sending the messages. The more active genes are the immune genes involved in the immune response. And we can explain these genes being less active because they correlate with the genetic associations known with autism. So it's where we know that there might be malfunctioning because of genetic changes. The immune signal can't be explained by genetic changes known in autism. And so again, is it something that's secondary to the neuronal abnormal neuronal signaling that is happening, or does it suggest an environmental response, something that we don't know, but we need to look into? And what it does tell us is that there is a degree of microglial activation in autism spectrum disorder, looking at many individuals grouped together. So what work has been done in people and humans? Not a lot, to be honest, but this is one early translational research that was done, study that was done, and they looked at maternal interleukin-6, which is one of the more common inflammatory cytokines in mothers during pregnancy, and then they looked at newborns, brain connectivity and functional MRI studies of their children at 2 years of age, and they found that you could predict the levels of inflammatory markers during pregnancy from the child's functional MRI results and their working memory results. So some of the clinical work and what does this look like perhaps in practice? So I don't know why that skips there. I'll just go back there. So this is a family that I met in Sydney. And what I should say, sorry, I didn't make it clear at the beginning, is that these are all genetic conditions, but what we're talking about are there important environmental factors as well. And so you can see this is one example of how this is genetic. You've got 2 brothers in one family. I'll just go back again. Their father had depression or has depression. Their mother has anxiety, depression, and chronic motor tic disorder. The 16-year-old has Tourette's syndrome and OCD. The 12-year-old has tics, ADHD, anxiety, ODD, and actually now has Tourette's syndrome too. And so, an additional factor, in addition to the genes there, is that mother also has an inflammatory condition with these bright red swollen plaques on her hands, and this is called sweet split syndrome, which is a neutrophil-led condition. Similarly, this family, this is an only child that I also looked after in Sydney with abrupt onset OCD at 4 years of age. And interestingly, she doesn't have any family history of any neuropsychiatric or neurodevelopmental problems. But her mother, they're both Fijian Indian, and her mother has total body vitiligo and Hashimoto's thyroiditis, and the daughter actually went on to develop vitiligo too. Raising the question, is there a potential linkage here? And so this was looked at to an extent by Dr. Tanya Murphy over in the States, and she looked at the maternal history of autoimmune disease in children presenting with tics and OCD in 100 of their patients seen through clinic. And she found that the rates of maternal autoimmune disease was about 17% compared with the general population in the US, which is estimated to be around 5%. This was a population study looking at using Swedish national data that looked at the rates of autoimmune disease in probands and family members of probands with OCD in the top graph there, and tic disorder and chronic tic disorder in the lower graph there. And they found that autoimmune disease was more common in mothers and fathers, in first degree siblings, of probands with OCD, and mothers and fathers of probands with tic disorder and chronic tic disorder. But not necessarily, slightly higher than in mothers, but not statistically significant. The relevance of this is to show that it may all be more complicated again, not just the environment in utero, but there may be immunogenetic links between autoimmune disease and these neurodevelopmental disorders too. And so that data formed the basis for my PhD research, which really looked at 2 questions. What is the rate of autoimmune disease in mothers of children with tic, Tourette's syndrome, and obsessive-compulsive disorder in our clinic environment compared with healthy controls? So I did have a control group. And then we also had a positive control group of neurological autoimmune controls. And so these were children with autoimmune neurological conditions, such as, for example, multiple sclerosis. And just generally speaking, autoimmune diseases tend to cluster. So our hypothesis had been that children with neurological autoimmune conditions would have higher rates of autoimmune conditions in their families. And so we looked at 200 children with tic, Tourette and or OCD, 100 healthy controls, and 100 neurological autoimmune controls. The second question was, do the children with tics, Tourette syndrome and OCD and a maternal history of autoimmune disease have distinctive characteristics? Can you pick these children? Do they present differently because their mother has autoimmune disease? So to answer that first question, we found that the rates of maternal autoimmune disease in children with tics and OCD was around 30%. This compared with around 20% rate of maternal autoimmune disease in the neurological autoimmune disease group and around 12% in the healthy control group. And so the rates of autoimmune disease in the tic and OCD group, rate of autoimmune disease in mothers in the tic and OCD group was statistically significantly higher than in the healthy control group. This is just to show you that these factors, maternal smoking, medication in pregnancy, neuropsychiatric disorders in mothers, and assisted conception were also more common in our tic and OCD group, but controlling for all of these, you can see that there was still a correlation between autoimmune disease, which is what we're looking at here, and tic and OCD. So if the line crosses 1 simplistically, it means that there's no statistical significant difference, but to the right of 1 or higher towards 10 shows that the odds are higher. And so what types of maternal autoimmune disease did the mothers have? And so the most common autoimmune disease, as you would expect, because it's the most common autoimmune disease in mothers or in women of childbearing age, was autoimmune thyroid disease. And this was relatively similar in frequency between the 3 groups. The other groups here were very heterogeneous or varied, so celiac disease, inflammatory bowel disease, vitiligo, alopecia, many different disorders were represented there. But what really stood out to me was that psoriasis was significantly higher in the mothers of children with tic and OCD compared with healthy controls and neurological autoimmune controls too. And I'll come back to that later on. To answer the second question, do children with tics, Tourette syndrome and OCD whose mothers have autoimmune disease, do they have specific features? Is this potentially a slightly different disease, slightly different disorder? And so these are the clinical features we looked at. So the top 3 were our primary hypothesis here. We hypothesized that children with tic and OCD of mothers with autoimmune disease would have a more rapid onset. They might have an infectious or inflammatory trigger at onset, and perhaps they have a relapsing, remitting course or severe fluctuations with infection, thinking, is this an immune version of tic, Tourette, OCD? But what we found was that none of those features were associated with maternal autoimmune disease. And in fact, out of all of these things we looked at, there were only 2 features that were associated with maternal autoimmune disease, and that was a chronic progressive course and also relief by non-steroidal anti-inflammatories. And I just make a note here that this is a lot of factors to look at. So even though, and this was not correct, the significance was not corrected for multiple comparisons, which means that there's a possibility that these associations were due to chance, but that still they're an interesting observation to make. This is a rather complicated slide, but just another aspect that we looked at, and that was looking at the blood of the mothers with autoimmune disease to see which genes were more active in their blood, and commonly across the group. And as you can see here, the early immune response genes were. And then we compared them with what was published earlier, and I showed earlier that paper by Lennington, which genes were more active in the brains of children with Tourette syndrome. And again, it was more the early immune response genes that were more active in Tourette syndrome. So this is exploratory data. It's more just out of interest, but it suggests to us that perhaps it's the early immune response that where this link between the immune system and neurodevelopmental disorders might sit. And then because there were varied autoimmune diseases, and because of they're suggesting that perhaps it's the early immune response that's important, we went back to the data that we had prospectively collected, looking at other conditions that are associated with inflammation. And so, as you can see along here, these were anxiety and depression. So obviously, I'm not saying everything to do with a link with anxiety, depression is immune mediated. That would just be preposterous, but they are pro-inflammatory conditions. Asthma, infection, preeclampsia, gestational diabetes, and smoking. These are inflammatory conditions, and they were more common overall in the mothers of children with tic and OCD compared with the 2 control groups. And so my colleague, Dr. Valda Han in Singapore now, she went back and did a systematic review looking at all the studies that look at pro-inflammatory conditions and neurodevelopmental disorders, namely autism, ADHD, and Tourette syndrome. And overall, there is a tendency towards pro-inflammatory conditions being associated with these neurodevelopmental disorders. And so what next? This linkage between psoriasis and tic and OCD, I think, is very interesting, and it points towards a potential link between interleukin 17, which is one of the inflammatory markers in neurodevelopmental disorders. And so this is where we're going to look at next, moving on from my study. And it wasn't just us that found this link with psoriasis. This is a systematic review by Timothy Nielsen, who collaborates with us, and he looked at Australian data, looking at the prescription of stimulants as a marker of ADHD, and linked it with the maternal health database, and also found this link specifically with psoriasis, maternal psoriasis, and ADHD. Overall, I think psoriasis has come up in a number of population studies, but I think it's under-recognised because most people with psoriasis don't need to go to hospital, which is where a lot of the data comes from. And it's not something that obstetricians would generally think about. And so what is psoriasis? It affects 1 to 2% of the population. It's inflammatory, autoimmune, as mentioned, skin condition. It's possible that the prevalence is rising, and the first peak in incidence is around that young adult age group. We know that in Auckland, thanks to a study done by Dr. Stephen Lamb, that 53% of those who present to the Auckland services with psoriasis in New Zealand Europeans, 16% are Pacific Islanders, and 10% are Māori, so mirroring our ethnic makeup and affecting all ethnicities. And so this slide here just shows the pathogenesis of psoriasis in one slide, but it was really that Th17 that I wanted to point out. In this, there have been a number of studies that have looked at maternal cytokines like that interleukin 6 one that I showed you earlier, and neurodevelopmental disorders. But what hasn't really been looked at is this Th17, and that is a key inflammatory mediator in psoriasis. It's also been shown to be key in animal models. So Gloria Choi's group in the States really looked at the immune response in those mice, and at what point can you block the effect on the offspring. And it really looks like the interleukin 17A pathway is critical. And so this goes on to the study that we're going to be doing, looking at maternal psoriasis and looking at neurodevelopmental outcomes. So the objectives of our study will be to investigate the neurodevelopmental profiles of children born to mothers with psoriasis compared with mothers without psoriasis. whether serum concentrations of interleukin-17A and other cytokines during pregnancy and at delivery correlate with neurodevelopment at 2 years of age, and whether gene expression in the newborn, looking at the different ways that we can look at that, using different ways so that we can look at that, differs in children born to mothers with psoriasis compared with healthy controls, and whether that gene expression is associated with neurodevelopmental profiles too. Oh, something's happened to that slide. I'm sorry, I'll go back. So how we will look at that is we will recruit 100 women who are pregnant with psoriasis and 100 women who are pregnant without psoriasis, and measure all of their children's development at 2 years of age using the Bailey scale of development, which is one of the gold standard measurements really. And we'll particularly look at social emotional scale. And then in a subgroup of 30 mothers in each group, we'll measure their cytokines in each trimester of pregnancy and at delivery. And also we will store those mothers' bloods in the first trimester so that we can look at gene transcription down the line if we need to. And then in the newborns, and using cord blood, we'll look at the newborn cytokine profiles and also the newborn's gene expression profiles. So why is this important? We obviously do not want to make all mothers concerned with psoriasis that their children are going to have developmental problems. And we are talking about one factor here, but in the big scheme of things, psoriasis and autoimmune diseases are very common and they're treatable, and they're not something that people typically think about when they're managing, unless they're severe, think about when they're managing pregnancy. Autoimmune diseases, if they are important in neurodevelopment, as what I've shown you suggests, then managing them if somebody is planning pregnancy will be very important. And understanding the role of how maternal inflammation and neurodevelopment may affect neurodevelopment may show us target options for treating neurodevelopmental disorders in the future, may show us whether some children will respond to immune treatments and guide us in this area, in an area where neurodevelopmental disorders are very common and are very challenging to treat. On that note, I'd just like to finish with really acknowledging the Neurological Foundation, who have supported me through this work, and even right back when I first became interested in research 7 years ago, I had a project grant from them too, so I'm very grateful to them. And also Scott Graham from the Centre for Brain Research at the University of Auckland, who is supporting this study, and our collaborators in Sydney as well. Thank you very much, and I'm happy to take questions.